you are from the various branches of the biological sciences. So I have not selected any topic specific to either microbiology, zoology or botany, but I have selected the topic which will be useful to all of you. And being a teacher, many of you must be from a postgraduate teaching and some of you might be from undergraduate colleges, but either you are teaching at the postgraduate or undergraduate, you have to deal with the experimentations. So today we are going to discuss about the experimentations and experimental design. You all are familiar that being a biological sciences, we have to do our research, and this research is the experimental research. We have to do all the experiments in laboratory, or depending upon your field of expertise, you may have to do in a field saver. Those who are dealing in a botany department, now they have to do in a crop productions. In zoology department, you have to work at a field level, and in microbiology, either in a field level, industrial level, or at a laboratory level. So depending upon at what level you are working, you have to deal with the experiment, and that experiments are why we want to discuss the experimental design. So in today's my 1 hour of talk, I will be discussing what is the experimental design, what are the common terms used in the experimental design, characteristics of a good experiment design, basic principle of experiment design, and step of the design of experiments. Now this is a content what we are going to discuss today. So if we come to the first aspects, what is experiment design? So before discussing the experimental design, we have to know why we want to do this design. And design of experiment is a method of experimenting with a complex process. When we are working with a biological sciences, our process are more complex as compared to the physics or chemistry, because whatever the factors, external factors we can control, beyond that biological factors, either you are dealing with the plants, animals, or you are dealing with microorganisms, then their metabolic activity as well as their physiology is affecting, and many of these parameters we are not able to control, and process become more and more complex. So the experiment design is the formulating the experiment to deal with the complex processes with the objective of optimizing the process, where we can get the best productions, best response in terms of the product. Experiment design is also defined or also explained as a set of a control experiments conducted in a randomized order. Here, each and every word is important. That is the set of control experiment. So you are doing the experiment under the control conditions. When you go for the field level, then it is difficult to do the experiment under control conditions, and that's why we have to go for various types of the design that we will be going to discuss in the next lecture. And then you have to conduct the randomized order. Why we have to do the experiment in randomized order? Because if you do, suppose if you have to conduct 1 to 25 experiment, and if you do 1 and then 2 and then 3 and up to, if you go to the 25 in a sequence, then there possibility that when you are doing your first experiment, you are more fresh, you are more energetic, and if you are continuing your 25th experiment, due to your tiredness or whatever other reason, you may not be able to give your attention as much as you are giving with your first experiment. And that is a uncontrolled factor which is affecting your process, and we have to minimize this. And for that reason, the best way of doing the experiment design is to randomize the order of the set of experiment. Number of experiment required is dependent on the type of the design what you are selecting. A simplest experiment, many of you, which you are giving to your students at BSc level or at MSc level, that mainly we are discussing with a one parameter at a time. But one parameter at a time experiment is the simplest experiment which will not allow us to go to the optimum conditions which you can apply at an industrial level or at a field level. That is why we have to think about the number of the factors which are affecting individually as well as how they are interacting with each other. Now, what we want from a design of experiment or experiment design. So there are certain questions what we want to achieve. So we want to do the variable effects on the system. Whatever the parameter you are selecting and that parameter you are changing, then what is the effect of that parameter on your results? That is the main aspects of experimental design. Then the magnitude of the variations. Suppose, as for example, we are studying the effect of temperature. And if I am varying the temperature between 25 and 30 degrees, the effect may not be significant or noticeable. But if I study the effect of temperature from 10, 20, 30, 35, 40, 45, then definitely I will find the effect of variables on the response. So how any variable, they are affecting the system, that is also the another aspects of the design of experiment. And by doing this, to determine the optimum level for the variables, not of one variable, but of the variables. So when they are interacting, either they are interacting in a positive way or they are interacting with a negative combinations, then either beneficial effects or harmful effects, or it may be just a synergetic effects that we want to find out. And on the basis of this, we have to go for the optimum level of the variables and get insight how to manipulate the variables. As I told you that when you are changing the variables, then with what range you have to change the variables. So your response would be having the significant aspects. So from experiment, we want these 4 or 5 things. And due to this, you can study the effect of experiment on the results, which is useful to you, which is useful for the publication, and which is useful to the society. Then why we need the experiment design or design of experiments? Question arises, no, as I said that what we want to derive from the experiments, to derive any of these things, we require the proper designing of experiment. Apart from this, why we want to design experiments are very costly, and particularly in biological science, they are much or the more costlier as compared to the chemical or the physical science. And when you want to go to a production level, because ultimate aim of any experiment is to study and to give the product at a production level. So instead of repeating your experiment at a production level, if you want to conduct this experiment at a pilot plant level or laboratory level, then how the experiment design should be made. So whatever results, even if you are getting in a conical flask or whatever results you are achieving in the laboratory, same result or a similar result you will achieve in a pilot plant, and also you can achieve to the production level. Design of experiment is necessary to minimize the number of the trials, number of the experiment, to get the maximum amount of the outcome. If your design is faulty, if design is not proper, then you may conduct more experiments and you get less information. So here you are minimizing the number of experiments without compromising the integrity of the data, integrity of the analysis, and not compromising with any aspect which is left behind. So the experimental design will give you the idea so you can get the maximum optimum conditions, best optimum conditions with a best achievable response from your experiments. Common terminology which is used in experiment design, I am sure that many of you must be familiar with this, but those who are not familiar with this, and those who have not done the statistical design, or those who have not done the complicated and the complex design to achieve the optimum conditions. So some of the factors they are not very much aware of. So the first factor, the first term is the factor, a variable which the experimenter, means we, those who are doing the research, that any researcher who can change it as per his wish and will, and that particular factor or particular variable is affecting the response. So a variable which the experiment will vary in order to determine its effects on a response variable, that factor, that is known as factor. And when this factor is studied at a different level, as for example in case of pH, suppose you are studying at 5 pH, 7 pH, 9 pH or 11 pH, a temperatures 20, 30, 40 temperatures, or inoculum, or you can say the amount of the fertilizer you are adding in the field, 1x, 2x, 3x. So this variations in any factors, that is known as the level of the that particular factors. The third one is the response variable. When you are varying these factors at a different level, then what output you will get, which we are measuring and which is useful as a product formations or process development, and this particular variable is known as a response variable. The variable used to describe the reaction of a process to variation in a control variable, that is the factors, that is known as a response variable. Noise factors, which is very important. Those who are using sophisticated instruments, there one of the result will come the noise factor of this, and you have to nullify the noise factors to get the results which is only and only due to the independent factors. Response is a dependent factors which depend on what factors you are choosing and how you are manipulating that factors. On the basis of this, the response is going up or down. But to find out that whatever the change in the response is either due to the control factors or which is due to the some uncontrolled factor which we are not able to control due to the various factors, that is known as a noise factor, or that leads to a noise factors. An uncontrollable source of variations in the functional characteristics of a product or a process, which is known as a noise factor. And that is very, very essential. Otherwise, whatever the data you will get, whatever the finding you will get, that may be either it may be negative or may be positive. That is not an actual outcome of your experiment. It is not the actual outcome of your design experiment. But it may be a plus minus of your experimental variable as well as the noise factors. Effects. The change in the response variable that occurs as experimental condition changes. Suppose if I am changing the pH to a higher level, that is alkaline level, or if I am decreasing the temperature to a lower sides, and if the productions or if the process becoming faster or if it becoming slower, that is known as the effect of your variations in a independent factors on your response. Interactions. As I said that most of us at a college level and even in many experiment at the PG level also, we are doing one parameter at a time or one factor at a time, where there is no question of interactions. But we know very clearly that if temperature is increased and at the same time if pH is also increased or decreased, then that individual effect of temperature or pH is not only with the pH or temperatures, but they are interacting with each other, and whatever the total outcome due to the interaction between the temperature and pH, due to the presence of one organism, suppose you are doing this experiment in the field, then due to the fertility of the soil, due to the rainfall during that season, due to the wind, all these factors are interacting with each other, and that leads to a synergistic effects. and that is the correct outcome. And that is why it is very essential when you want to put your data on a production level, or if you want to develop your process for industrial or field level, you have to study the interaction. You cannot neglect. Interaction may be a two-way interaction, three-way interaction, or multi-way interactions, depending upon how complex experiment you are performing, how complex experiment you are designing. Now, repetitions and replications. Due to my nearly 40 years of job as a teaching, many times the students know they are confused between the repetition and replication. Repetition of any experiment and replication of experiment, both are different and they are done for a different purpose. Repetition is a multiple result of a treatment. Here, most important is a individual single treatment. When you are a same treatment, you are taking the many samples from the same class, which is known as a repeated examinations of your results. And or a same class you are running to several times. Running several samples during a one experimental setup, which is known as a repetitions. And replication is duplicating the entire experiment. Suppose your experimental design, which you are having a 10 set of experiments, and when you are doing all the 10 set of experiment twice or thrice, it means you have replicated 2 or 3 times. And in any of the authentication data, we have to replicate minimum 3 times. 3 replication is must. And you ask yourself, particularly in biological science, we are avoiding these replications because our experiments are costly, it is taking longer time, and many times we are not replicating. But if any experiment which is not replicated for 3 times, then you cannot rely upon the results what you have achieved for that experimentation. Randomizations, what we have discussed in the first or second slides, technique used to randomize the order of the experimental run or the assignment of the experimental unit to the different factor level or the combinations. That randomizations is required even in case of sampling, in case of setup of an experiment, in case of the performance of experiment in particular sequence. Because if you are not doing the randomizations, then your personal bias will come into the experiment. And the personal bias would be responsible for your choice or liking or unliking. And that liking or unliking is not allowed in case of the science. Because it's not my liking. It is the science, what we have to do. And that's why randomizations is very important. Residual, the difference between an observed results and predicted values. When you are doing a statistically designed experiments, and if you study this experiment in a computer design experiment, when you will analyze the data, you will get your observed response as well as the computer will get what are the predicted value. And your observed value, they are how much different from the predicted value, that is very important. If predicted value and observed value are not matching with each other, then it means your modeling system or modeling equations may not be accepted. So a residual value and the observed value may be as close as possible. Next terminology in case of experimental design is run or trial. A single setup in a design of experiment from which data is gathered. For example, a 3 factorial experiment, then you are doing 2 raised to 3. It means you are making 8 experimental setup. So this 8 experiment, they are known as a run or they are known as a trial of experiment. And the application of one treatment to one experimental unit. The application of a one treatment to one experimental unit. So when you are having a 3 different factors studied at 2 different levels, and if you make the combinations, you will get 8 combinations. So that 8 combination, then it is known as if one trial is over or one run is over. Treatment combination, that is very much similar to the run trials. Now factor into level, suppose as I said is a 3 factors and 2 level, so 2 raised to 3, 2 into 2 into 2, so you have to go for 8 experiment. Suppose 2 factors and you are going 2 level, and if you are going for 4 factors, then 2 raised to 4. So it leads to a treatment combination of a 16 experiments. Significance. We are very much aware of when we are doing the experiments, we look to the p-value or 95% significance, 99% significance, or 99.9% significance. This is very important for the experiment as well as being a teacher. When you are judging the result of your student, examination results, and if you are not going for the significance level, and if we give more or the less marks, then student is at loss. So we have to find out, even if you repeat this experiment, due to the internal variable which you cannot control, no, there would be some fluctuation in the result. And that fluctuation in the results, we have to take into considerations that what plus or minus result any student or any researcher will do, or if the same experiment is conducted in another laboratory, what fluctuation they will get. Or if yourself you are repeating this experiment for 100 times, then what are the chances that the desired result you will get in how many experiments? Aliasing, another terminology which is used in an experimental design, when any 2 factors or interactions which are set at an identical level throughout the experiments, then it is known as these 2 factors or these 2 interactions, they are aligned with each other. And confounding factors, another terminology which is very essential to understand before designing the experiments. The experiments are considered to be confounded when their test profiles contain the same pattern of the test setting, making it impossible to evaluate the 2 factors independently. So when the effects, you cannot find either effect is due to A or B, or how much is effect due to A, how much effect is to B, then that is, we can say, due to the confounding effects during your experiment. If we know the more detail about the confounding factors, then confounding factor is a hidden variable, which is not observed during the experimental process, which is not known to us. And if you will overlook it, whatever the result you will get, that is, results are not acceptable. And in a statistical or a research model that affect the variable in question, means the variable what you want to study, but it's not known or acknowledged, which is known as a confounding factors, which disturb your results, which disturb the data, and the data are not scientifically true. This hidden third variable causes the 2 major variables to falsely appear to be correlated or to be in a causal relationship. Next factors what we want to study, effect of, as for example, coffee drinking, lung cancer, and the same person is also smoking the cigarette. So it is very difficult. You cannot say the cigarette smoking is responsible for cancer, or a coffee drinking is responsible for cancer, or a coffee and cigarette smoking may be interacting with each other, and which leads to more chances of the lung cancers. No, this type of the situation when arise, that is known as a confounding factors. If a confounding factor is recognized early enough, adjustment can be made so confounding factor does not disturb our result, does not spoil our result, and you will get more authentication, more authentic results. Now discussing with the various factors, what is the experiment design, then if we come to know, or if we want to find out what is a good experiment design, or which design we can say it's a good experiment. It is very, very difficult. It is very hard to define a good experiment. However, when we want to have a experimentations, then a good experiment is an experiment that tests what you want to test and control for everything else. So there are so many other factors in a good experimental design, but in the summary, if we say a good experiment is an experiment which can test what we want to test and control all other factors which are affecting in the experiments. A good experiment estimates the effect you are interested in and minimize the confounding factors or minimize the experimental error, minimize the error due to the noise, minimize the error due to the uncontrolled factors. It means what is your intentions? So good experiment is an experiment which give you the estimation of your intended variable, intended experiment setup and minimize the other factors which we are not able to control or what we are not able to estimate. Then a question arises, what is a bad experiment? Or what is a bad experiment design? I will give you the one example for this. Suppose if you want to study some medicines or a drugs and you have selected 20 patients for the experimentations. Out of this, if the drug is given to a 10 male which are having the age between 50 to 60 and a blank or placebo is given to a control group, but there due to the faulty design they are selecting instead of male 10 female. So even if you select a 10 male 50 to 60 age for a drugs and if you select a 10 female for a placebo for this design, then also it is faulty. And here one more factors you are having the difference. The age of the male is 50 to 60 years while the age of the female what you have selected is 20 to 30. So the physiological conditions due to the aging, physiological variations due to the male and female effect of drugs are variables. As we know that in the COVID-19, now when we go for the third phase of the testing, now you have to test a wide variety of the people, various people from the various state, people from the various country, people from the various continent. Then the results are applicable on a planet Earth. So and that result should be to find out what would be the change in the effect of whatever the vaccine we are going to give to the male or female either from India or America or Russia. Then as the physiological conditions, metabolic conditions, the type of the food what we are taking, no, that would be different. So a bad experiment, when you are not taking care of all this part, then your experimental design is not successful. When mutants were considered with a wild type of the organisms, when you want to compare, then mutant should be compared with mutant or a wild type which will compare with a mutant with a control factors. So you can know that what is the effect of your variable on wild type of cell and what is the effect of your variable on a mutant. It is not that you can study the one variable on wild type of the organism and second variable on the mutant organism. If one group is tested in one lab and other group is tested in the second lab, that is also a bad experiment design. If you want to do this, if you are not able to conduct this all the experiment in the lab 1 or lab 2, then randomly you have to divide the number of trials and the test, and randomly some of the experiment should be done in lab 1, other experiment should be done in lab 2. Some of the replications should be performed in lab 1, and similar way the replication and repetition should be performed in lab 2. So when two experiments are replicated and they are repeated in a two different lab, and if the results are slightly or different, then that factor you have to find out that this factor is not due to your experiment, but that factor is due to the choice of a two different lab. Problem of a badly designed experiments. When experiment is not properly designed, and which happens many times, even Many of you must be a PhD guide, and when you are guiding your students and they work for 3 or 4 years, when they start writing their thesis, then we find some problem, such as, and that is due to a badly designed experiment. Which conclusions can we draw from such experiment is a question mark. When we are not able to conclude, or when we are not able to draw the conclusions after 1 year, 6 months, or 2 years of work, and that is not the fault of the experiment you are doing, but that is the fault of the design of experiment what you have selected. So experimental design, you have to think much prior before you are starting your experiments. Can we answer the question of the interest? Once again, this question mark. If it is a not properly designed experiment, you cannot answer the question of your interest. Suppose if I want to find out either I am audible to you or not. If I am not having any criteria to listen the response of the participants, or if your mic systems, I don't know in which way it is arranged, then I may not able to answer. Can we generalize our conclusions? Whenever you are doing your experiment, that conclusion may not be only specific to you. The conclusion should be when this experiment is done in any other lab, experiment is done in any other state, or even experiment is conducted in some other country, you should get the similar conclusions. Any responsibly skeptical persons, or a doubtful person, or a critical person would doubt the validity of whatever conclusions would be drawn from this type of experiment, if it is a badly designed. For some bad experimental design, there are no statistical fixing. After the end of your experiment, you try to apply the statistical procedures to analyze the data, to perform this experiment, then there is no statistical fixing is available. What is required for good experiments? As we said that good experiment is necessary. We have also discussed the complications and the drawback of the negatively designed experiments. So what is required for a good experiment? Once you know this, then you can go for the good experimental setup, or you can go for a good experimental design. So the first and foremost requirement to go for a good experiment is well-defined one or more hypothesis. Any experiment, many times, when I examined the PhD thesis and this, there was no hypothesis written in the first chapters or in the second chapter. Even when we ask at the time of the Viva, students are not very much clear that what the intention was. What was the hypothesis? Either hypothesis is proved or disproved. So for a good experiment design, you have to formulate a well-defined one or a more hypothesis. Mutated bacteria reacting differently than a wild type of bacteria to stress condition. Now this, when I am studying with a wild type of organism and I am studying with a mutated organisms, as for examples, yesterday, in case, they have said that in UK, new strain of the COVID viruses has developed, and they have got some increasing number of the COVID patients with that new mutant strains. And due to this, UK governments are thinking to lock down for this Christmas. And even the president of UK, he is very much worried. And so this is what happening, that mutated bacteria reacting differently than the wild type of bacteria. So this new mutant virus is more pathogenic, more virulent as compared to whatever throughout the planet we are suffering from the COVID viruses. If it is there, and today even in India also, particularly the Kejriwal, he has requested central government to ban tourists from the UK, Britain, because this new mutant virus is found in UK and any person will come from the UK, and if that virus will come in India, then you are finding that difficult. Second question is, how do change in the environment which influence the performance of a selected culture? So this is simplest hypothesis. So when you design your experiment, you have to frame the simplest hypothesis. And characteristic of good experiment design, the experiment should provide unbiased estimate of a process variable and treatment effects. Here the word unbiased is very important. It should not have, as suppose being I'm a microbiologist, if I'm doing this experiment in a field or if I'm doing this experiment in animal, I want to prove that my microorganism is much effective as compared to the physiology or metabolism of a plants or animal. It is my bias. So a good experiment design should be conducted in such a way so there is no bias. A bias of the experimenter will come into the pictures. When you are processing, when you are selecting the variable, when you are testing the effect of these variables on the various experiments. The experiment should provide the precision necessary to enable the experiment to detect important difference. Here, the word precision, accuracy, and precision is very important. Reproducibility, reliability. I think there are so many terms, and you must be familiar with this. Reliable experiment. What do you mean by the reliable experiment? Now, when you are reading many of the WhatsApp which are going nowadays regarding the either we have to take the Sputnik vaccines or Pfizer vaccines or vaccine produced from India. So that is depend upon the reliability. Which vaccine is reliable? And reliability of vaccines or reproducibility of the result depend on the precision, in which with what precision experiment were conducted. So precision is necessary to enable the experiment to detect important differences. And that differences should be precisely formulated, precisely differentiated, and precisely explained. And to do this precision in the decision making, you have to perform your experiment more precisely. The experiment should plan for the analysis of the results. That should have a proper planning that how this that result would be designed. Either it can be explained in a one experiment at a time, or it can be explained in terms of a factorial experiment, or which particular software you can use for analysis of this result. Accuracy and the precisions. I have tried to show this. What is the accuracy and precision? Now, from this data, now suppose this first figure where 7 different dots are shown. Now this is the center points. So we can say this particular experimenter, the way he is doing the experiment, now is neither precise nor accurate. It means the results show that the experimental design and the results are low accuracy and the low precisions. The second experimental results, where all the results, they are falling at one particular level, and this is happening in many of our students' results, in many of the PhD thesis results. Students are very happy. Now, but this we can say a high precision but low accuracy, because we want the results in the center part of this. And all the result means the student might be having some confounding factors, maybe a fault in the instrument, maybe the fault in the reagent they have produced, anything. And due to this, he is getting the precisions, but it is not accurate. The third experimenters, where he is having high accuracy but low precisions. Now, when the results are coming to the whatever the point which is decided, but still they are scattered. They are scattered into outer surrounding. And this is the best thing, high accuracy and high precision. So I think with this one figure, all of you must be very much clear that we require accurate and the precisions. It is not allowed that if you go only for accuracy and without precisions, or if you go for precisions without accuracy, then your results may not be accepted. Now, this is what happens in terms of the high accuracy, low accuracy, and what type of the graph you will get. What type of the graph you will get, and this will get the very steep glass, very wide glass. Then you will get the skew on one side, and this indicates the precisions and accuracy and that importance. The experiment should generate results that are free from the ambiguity of the interpretations. You have to conduct your results, you have to conduct your experiment in such a way so obtained results have no ambiguity. No ambiguity, and when you are interpreting your results, many times we find that where we have not added anything, that experiment will give the more response, and when you are increasing the dose up to certain level, you will get the increase in the response, then suddenly decrease, suddenly increase. So that is the ambiguity. Experiment should point out the experimenter in the direction of improvements. Your experiment should be designed in such a way so it point out that how you can go for path of steepest ascent, how you can go for the optimization, which provide the direction of the improvement in the experiments. The experiment should be as simple as possible. Even if you are dealing with a complex data, you are dealing with a multiple factors at a multiple level, but you have to choose the experiment as simple as possible, but you are not compromising with the accuracy, we are not compromising with the precision, you are not compromising with your hypothesis, but experiment should be simple in terms of easy to set up and carry out. The experiment should be simple to analyze and to interpret. Its samples simple to communicate or to explain the others. Many times, now when you are doing the experiments and you are explaining to other persons, you may be the expert of your experiment, your field. All the other persons, they are not expert. So your explanation should be also convenient and they can be easily understood by the different persons. What decisions are required for planning the experiments? So, how many minutes more I can take? What measurement you have to make, that is the response. What condition we have to study, that is the treatments. And what experiment material we are using, that is the unit. So, what decisions are required for planning the most experiment is response, the treatments, the units. And before starting your experiments, you should be very much aware that what you have to do, where you have to do, how you have to do, what are the things which is required for this. Now, many times when we are going for the RDC and when we are telling students to give the title of their PhD work, then we ask, do you think that this experiment is possible in whichever college or whichever university you are studying? Do they have this type of sophisticated instruments? Even if you are having that instrument, do you have that much money to purchase the chemicals? Or the third thing, what we ask, do you think that you are able to complete this experiment in 3 or 5 years? So these are the things which is very essential, which you have to design, which you have to ask before you start your experiments. Simple rule for developing the best experimental design are clearly identifying the objective of the experiments and response variables, less flexibility in designing, including the definitions, outcome and analytical modes, decrease amount of the bias towards the research findings, clearly identifying the scope of the inference, and define a simple unit as independence. So if you follow this simple rule for defining the experiment, clearly identifying the objectives. What is the objective of your experiment? That you have to find out. These are 10 simple rules for developing a best design of experiment. So we will discuss this, and later part we will discuss in a second lectures. So first, your objective should be clear. Then your experiments are not much flexible. It should be stringent. If more flexibility, then there are more chances of error, including the definitions, outcome, and analytical model what you are using. They should not be very flexible. Decrease amount of the bias. No personal bias, no institutional bias, no bias from the field should come. And for that, the thing what we have discussed is the randomizations of the experiments, clearly identifying the scope of inference. What analysis you are going to derive from this? What is the scope of your experiment? What is the scope of your research? That is clearly identified. Define a sample unit as independent. No, first thing we'll start with the sampling. When you are making the compromise in the sampling, many people, they don't know either it is a randomized samples, unbiased samples, or strategic samples, or a selected samples, and that create problem in the experiment design. Having an appropriate amount of the samples and sample units. When you are doing the survey, then sample should be in terms of hundreds and more than hundreds. But if you are having too many samples, then the cost will come into the picture and difficulty will come into the analysis of this. Avoid pseudo replication in the experiments, which is not really replications. Researchers should not limit themselves to only analyze a p-value. As I said, the 95%, 99%, 99.9% that p-value. Now people are just crazy behind the p-value, and they say my experiment is significant at 95%. My experiment is significant at 99%. And many person, they will say my experimental results are significant at 99.9%. Now, you must have come to know that in the COVID vaccines, they say if it is effective 5 to 60%, it is acceptable. Resource availability. What types of the resource are available, that we have to see. Look into the previous research done. Many students, they are not doing the literature survey. Many guide, they are also not much familiar with the literature survey. And at the end of 2-3 years, when they will start writing the research paper, or when they start writing the PhD thesis, they find that this experiment or this type of result was reported before 5 years and 10 years. You may not be able to publish your paper. So that previous research, literature survey is very much essential. I think with this we end. Now there are more slides, but we go to the next lectures after some time. If you have any questions.